All right, guys, you should have just finished your 4.3 notes over protein synthesis. So this is just going to kind of simplify the process into the application portion. So first thing we need to note is we can make proteins, or protein synthesis can happen during any part of interphase. And remember, interphase is 3 steps. So the 3 steps of interphase are G1, S, and G2. All right. Okay, next. So before we start filling in our bases, let's just quickly review what you did in notes talking about protein synthesis. So protein synthesis is 2 parts, right? The first part that we are going to talk about is when DNA is transcribed into mRNA. And remember that M stands for messenger because it carries the code. Okay? This process has to happen in the nucleus because it involves DNA. So DNA, remember, cannot leave the nucleus. So step 1, transcription, where we make mRNA, happens in the nucleus. Step number 2, known as translation, is when mRNA that we just made in step 1 is translated to a protein in the cytoplasm at a ribosome. Remember, that's the organelle that helps with protein synthesis. Okay, couple reminders that we want to get down before we start practicing our 2 steps of protein synthesis are the letters that we can use, or the nucleotides that are possible in DNA versus RNA. So remember, in DNA, the 4 different nucleotides that we can have are A, G, C, and T. Okay, remember A pairs with T, apples go in the tree. C always pairs with G, cars go in the garage. When we switch to RNA, what we learned from our notes is we can still use A, we can still use G, we can still use C, but anytime we would think we would need a T, we are going to have to use a U. That U stands for uracil. Okay, that is our new nucleotide, our new base. This is the one that allows RNA to be able to leave the nucleus. Okay, so when we are looking at our first step, so when we're transcribing to mRNA, that's where we're using those base pair rules. So if you look at your first one, we're going to use the template strand of DNA, and we are going to match our letters. So remember, T pairs with A. A, now that we're in RNA, pairs with a U, and C still pairs with G. Okay, moving on to our next codon, our group of 3. C pairs with G, A pairs with U, and T still pairs with A. Next 3, G, G pairs with C, A pairs with U. Then we have A, C to match with that codon. And our last one, G pairs with C, A pairs with U, A pairs with U. So we've done part 1, or step 1, where we go through and we transcribe our DNA into mRNA. Okay? Now is where we go to part 2, where we're going to translate them into a protein. So we need to figure out which amino acid goes with each of our codons. Remember, each group of 3 is called a codon. Okay, in order to do this, that's where we get to use either your chart or the wheel. Okay, so remember, you have both options. The wheel, you start in the middle and you work your way out. If we were using this chart, okay, I would find my first letter, which is A, okay? So my first letter is A. My first letter is A over here. Then my second letter is U, so I go up to the top. So I want A, U, so I'm in this box so far. My third letter I go over is G, so if I follow it across, my first amino acid is MET. Okay, and we always, or we can shorten these to write them with the first 3 letters of that amino acid's name. Okay, so let's go ahead and try the next one. So GUA, again, first letter, G, second, U, third, A. So if I follow all of those over, GUA gives me VAL. That is my amino acid. Third one, if I go from there, okay, I've got G, C, and then U. So if I follow it, GCU gives me ALA. Okay, 2 more before we complete this protein synthesis strand. I've got AAC for the next one. So A, and then C. So if I come across, AAC gives me ASN. And then for my very last one in this section, I've got CUU. So CUU. CUU gives me LEU. All right, if you flip the page, I'm going to show you your application questions in practice. You could do those same steps, but using your codon wheel, and then you can decide which one you prefer. So if you remember, our first codon was AUG when we were looking on the previous page. So if I want to use the wheel instead of the chart we did on the previous page, I would start in the middle. Okay, my first letter is an A. So of the first 4 letters in the center, here's my letter, A. Okay, my second letter is a U, so I need to look at these 4 that are attached to that A, and I need to find the U. So far I've gone A, U, working my way from the middle out. My third letter is a G, so then I see these 3 or 4 that are attached to that letter U. I pick the G. Okay, so I've gone A, U, G. I've made my way out to my amino acid's name. So my first amino acid is MET. Remember, you can shorten it. Just write the first 3 letters instead of writing the full name every time. Okay, what I need you to do now is go ahead and look at the application questions and practice. So you've got 3 on this page that we're looking at now, and then you have 2 practice problems where you're transcribing and translating to figure out the correct amino acid sequence. When you are done, raise your hand. I will come check them, and I will hand out your Yeti Genetics assignment.